now that the doors are closed, let's go ahead and get started. Good morning. This is session 301, Biofuels of the Research and Innovation Symposia. My name is Krishna Pagila. I'm a professor at the Illinois Institute of Technology here in Chicago. So we have all three speakers right here, and our alternate is also here, and the alternate paper is in the proceedings. So we start off with our first speaker, Shashwat Vajpayee. He's a PhD candidate at Columbia University in the group of Karthik Chandran. Karthik couldn't be here, but Shashwat will take care of the presentation, I believe. He's going to talk about conversion of food waste into biodiesel through sequential fermentation and enhanced microbial lipid production and novel platform technology. Shashwat.
Good morning, everyone. So today I'm just going to present our work on resource recovery, on recovering all the carbon present in organic waste and its microbial conversion to lipids and biodiesels. So one of the major challenges in the global health today really is the lack of adequate sanitation. The areas shown in red on the map represent about 50%, more than 50% of the world's population, and this population still does not have access to any kind of improved sanitation facilities. And the rest of the countries in the world have some sort of sanitation with vastly varying degree of applicability and success. Now the other challenge to the global health in the recent times has really been the food insecurity due to the rise in food prices. And in the developed world at least, this rise in food prices can in part be attributed to the rise in global biofuel production. Since almost all of the existing technologies today depend heavily on the agricultural commodities, they inadvertently contribute to the rise in food prices, either directly through use of food crops like corn and soybean oil for biofuel production, or indirectly through use of agricultural land masses to provide feedstock for biodiesel production. So today, there is a very critical need to develop sustainable and innovative technologies which can address these issues in a concomitant fashion. Technologies which can provide sanitation, but also recover all the valuable resources present in the waste and use those resources to supplement the world food and energy needs. So this is the current fate of waste, different waste streams around the world. So for the most part, sadly, there is no treatment. The waste just ends up in landfills or even worse, into the natural water bodies like rivers and oceans. And then there is increasing degree of sanitation. Some people just use waste directly as fertilizers. Some just treat it to remove the pathogens. In some parts it's simply burned. And in the developed world, where we have sophisticated technologies like anaerobic digestion and other sanitation technologies, but these technologies usually require a very massive inputs in terms of energy, in terms of effort, and capital inputs, and different chemicals to provide sanitation, and this makes them unrealistic for the undeveloped or the developing world, which lack either the technical expertise or the economic resources to develop such systems. So now the question we ask ourselves is, talking about just sanitation enough, or is there something else that we can do with the waste? Is there a way to recover all the resources present in the waste and produce multiple products? Go to multiple endpoints? Can you provide clean water and produce biofuels from that waste? I know resource recovery is much talked about, but even today, to this day, no technically sound, environmentally sustainable, and economically viable technologies exist which can address these issues. So our work represents one of the first technologies which is directly applicable to the existing systems around the world. So based on these broad goals, our specific objectives of this study were, first, to develop a fermentation platform. And this fermentation platform sits at the confluence of organic wastes from diverse sources, may it be food waste, domestic waste, municipal solid waste, fecal sludge, virtually any kind of waste imaginable. And what this platform does is it recovers all the COD, all the carbon present in that waste, in the form of volatile fatty acids. And since it removes all the carbon, it also provides sanitation. And the next objective then was to develop a biomass-based technology which can then take those volatile fatty acids and microbially convert them to biodiesel precursors or lipids. Now this is a brief overview of our process of how we achieve those goals. It all begins with organic waste. Again, like I said, any kind of organic waste imaginable from diverse sources. And then all the organic waste then enters the fermentation platform where it is converted to volatile fatty acids. All the carbon present in the waste is converted to volatile fatty acids. And how do we do that? We basically achieve that by cutting short the process of anaerobic digestion. Instead of waiting with a 15-day SRT to produce biogas, we cut it short. We stop it at 2 days or 3 days at this process of acetogenesis, where all the carbon is converted to acids. And this way, we are able to get very high concentrations of volatile fatty acids in the effluent streams. And then, these volatile fatty acids make very good carbon source for culture of oil-producing or oleaginous microbes. The oleaginous microbes are basically cells or bugs which have the capability to accumulate very high percentage, sometimes up to 65 to 70% of their cell weight in the form of lipids or fats. And one such oleaginous yeast, the one which we have been working with, is Cryptococcus albidus. So in the next step, we convert all those BFAs into lipids, which are the biodiesel precursors. And then the cells are harvested, the biomass is harvested, lipids are extracted, and then chemically converted to biodiesel via a simple transesterification reaction. Now the third step of this process, the microbial conversion of BFA to lipids, is really the heart and soul of the process. It's the pivotal step which determines the feasibility and applicability of this process. So most of our further research was focused on maximizing the lipid production using the yeast. Now the lipid content of the cell depends on many factors. It depends on carbon concentrations, different carbon sources, and even the compositions of the carbon sources. Then it depends on different nitrogen concentrations. And not just the individual concentrations, but also the relative concentrations of carbon and nitrogen, or the COD to N ratio. And then it is also affected by different reactor configurations, a batch reactor or a chemostat reactor. So we tested all these different factors, all this whole spectrum of different possibilities. We tested them in our lab. We started with different carbon sources, operating a lab scale fermenter on food waste procured from the cafeteria. We produced high concentrations of volatile fatty acids. And then we analyzed the composition of those volatile fatty acids, and to see how they match up against the pure BFA or synthetic BFAs, we mixed the BFAs, pure BFAs in the same composition and used those as the carbon sources. Then we tried different initial BFA concentrations going from 2 gram per liter to 8 gram per liter. And then finally, we used glucose as the carbon source to provide a benchmark really, to establish an upper limit on the maximum lipid production potential of C. albidus. And then using fixed concentration of BFA and ammonia as the nitrogen source, we tried different COD to N ratios. Going from far excess at COD to N of 5, we progressively decreased the nitrogen concentration to excessive limitation, extreme limitation at 250. So we tried these 5 different nitrogen concentrations, going from far excess at 5 to excessive limitation at 250. And then we tried all this in a batch reactor and a chemostat. But due to time constraints, I think I'll just show the results from our batch reactors today. So allow me to move on to the results part. Let's first look at the effect of nitrogen concentration on the biomass growth and the lipid content of the cell. It was evident looking at the results that going from excess nitrogen to limited nitrogen caused a slight decrease in the biomass concentration. But this decrease was accompanied by a very sharp increase in the lipid content of the cell, going from 19% all the way up to 28%. However, once the limiting concentrations of nitrogen were established, any further nitrogen limitation, any further increase in COD to N ratio did not have an effect on the lipid concentration, did not have a statistically significant effect on the lipid concentration or the biomass concentration in the reactor, and it remained constant. Now let's look at the specific growth rate, maximum specific growth rate of the cell. It behaved in an exactly opposite manner of the lipid content. Going in the limiting nitrogen concentrations, further reduction in nitrogen concentration kept on decreasing the maximum specific cell growth, going from 0.04 to 0.02. But going the other way and increasing the nitrogen concentration, did not increase the specific cell growth rate, and it remained constant. Now let's also look at the effect on different yield coefficients, the COD and nitrogen-based yield coefficients. So the plot on the top, this plot, is basically the effect of different nitrogen concentrations or different COD to N ratios on the specific biomass yield, the biomass produced per gram of COD consumed. So it shows that going from far excess to limitation did not have an effect on this number, and it remained constant. This means that cells were able to take up the same amount of COD regardless of the nitrogen concentration present in the stream. Now, we should note, however, that X represents the biomass, which is the sum total of the cell weight and the lipid present inside the cell. So what we understand is from this figure is that even though the cell was taking up the same amount of carbon regardless of nitrogen, it was just channeling it to different endpoints. Under excess nitrogen, it was producing more biomass, but under limited nitrogen, it was channeling all the carbon to lipid production. And contrary to this observation, the effect on the yield, the nitrogen-based yield coefficients, biomass produced per gram of nitrogen consumed, it kept on increasing, going from 5 to all the way to 35, moving from excess to limited nitrogen. And this means that under nitrogen limitation, cells are just able to assimilate all the nitrogen much more efficiently than the excess nitrogen. So the process implication of this result is that even if there is high variability in terms of nitrogen concentration in the influent stream, we can still expect a fairly steady biomass concentration in the reactor and therefore a fairly steady operation. Now, to understand this effect of nitrogen on the lipid content and the yield coefficients, we need to go down to the metabolic level and look what pathways nitrogen affects. How does nitrogen change the lipid concentration of the cell? So, the citric acid cycle is really the key metabolic pathway inside the cell under aerobic conditions. It provides many high energy molecules like ATP, NADH, and precursors for the synthesis of many other important molecules required for the growth. And one such molecule is alpha-ketoglutarate. This molecule is the key molecule in the synthesis of protein inside the cell. And therefore, when cells have high nitrogen concentration, it can grow, it can make a lot of biomass, and to do that, it needs a lot of protein. And to make a lot of protein, cell needs to make a lot of alpha-ketoglutarate. And under those conditions, the citric acid cycle is moving very fast, and therefore it is also producing a lot of high energy molecules like ATP and NADH. And that's why we observe the high cell growth rate, since cell is in a very high metabolic state where high nitrogen is available. Now, what happens when the nitrogen concentration goes down? Cell cannot make any more protein, so it does not need any more alpha-ketoglutarate. And therefore, the citric acid cycle comes to a screeching halt, it stops. And since the citric acid cycle stops, cell does not have any more high energy molecules, and it enters a lower metabolic state where cell is not growing very fast, not making a lot of biomass. And what happens under these conditions is that since the carbon is still present in excess, we provided excess carbon, cell is still producing acetylcoenzyme and it's also producing isocitrate. It's also producing a lot of isocitrate, and under those conditions, then the isocitric acid is taken away from the citric acid cycle, it's transported outside, and it's converted back to acetylcoenzyme, and then it is assimilated in the form of lipids or other carbon storage compounds. And this is the reason why we saw, moving from excess nitrogen to limited nitrogen, there was a very sharp increase in the lipid content of the cell. Now let's move forward to the effect of different carbon sources and different carbon concentrations on the cell. So we compared the pure BFAs with the fermenter BFAs. Under the same carbon and nitrogen concentration, cells were able to produce almost similar biomass, but looking at the lipid content and the cell growth rate, it decreased in the case of fermenter BFAs. And this is possibly because of the presence of other fermentation products in the effluent stream, which might have been inhibitory to the cell growth. And since we can go back to our fermentation platform, the objective one, and since this process of the fermentation is highly customizable and very flexible, we can easily address this issue by changing the conditions of the fermentation and producing a more suitable VFA feed. Then we also tried different VFA concentrations. from 2000 to 5000 and 8000 milligram per liter. At 2000 milligram per liter, the carbon concentration was too low for the growth compared to nitrogen concentration, and therefore the cell growth was and the lipid accumulation was limited by the carbon exhaustion of carbon rather than the exhaustion of nitrogen. And then we moved to a very high concentration of 8000 milligram per liter. So at this concentration, we observed that VFA exhibits substrate inhibition in the cell. And since it has been reported in other literature that acetate tends to interfere with the transport of ATP across the membrane, and this is the reason why we did not observe any growth at 8000 milligram per liter. However, at 5000 milligram per liter, we produced very high biomass and cells were able to accumulate high concentration of lipids inside them. And therefore, we chose 5000 milligram per liter for rest of the studies. And now let's look at the glucose numbers. Well, as expected, the biomass and the lipid concentration was very high on glucose. Comparing with the BFA from the fermenter, the lipid content and the biomass was almost 1/3 on the BFA from the fermenter compared to the glucose. So this makes glucose the substrate of choice from a technical standpoint. But when we look at these numbers from an economic standpoint, the cost of glucose in the international market is $500 per ton compared to the cost of production of BFA, which is $30 per ton. So the final cost per ton of the biodiesel produced from this process is going to be much, much lower using BFAs. And not only the BFA concentration, but different BFAs also affect the lipid content. So we tracked the consumption profiles of the cell, consumption profiles of different acids in the batch culture. That's what this plot shows. Acetate, propionate, and butyrate, which were present in our BFA mixture, were taken up in different manners by the cell. Looking at the acetate, it went down very sharply, and by the time cells reached the stationary phase, acetate was all gone. And then cells started consuming propionate. And at the end of fermentation at 96 hours, the only acid still remaining is butyric acid. It wasn't consumed at all by the cells. So what this tells us that we can go back to the fermentation platform we developed and increase the HRT slightly or modify their conditions to get a predominantly acetate in the BFA effluent, and which can possibly further enhance the lipid production. Now let's look at the fatty acid composition, how the different nitrogen concentrations affected the different fatty acids that were present in the lipid. So not just the lipid content, but also the lipid composition, it has a very profound impact on the quality of biodiesel that can be produced from it. If all the fatty acids used is saturated, like C16-0, which is basically a 16-chain carbon with no double bonds, all single bonds. So, and we know that saturated fats tend to be solid at room temperature. So a biodiesel produced from mostly saturated fats will have very poor cold flow properties and will tend to gel at lower temperatures. And therefore, to be used as a fuel, we will need to make higher blends of this biodiesel with petrodiesel. Now going to the other end of the spectrum, if we have a lot of unsaturated, the polyunsaturated fatty acids or 2-PAS as our substrate, like C18-2 and C18-3, linoleic and linoleic acid. In that case, since these fatty acids are highly prone to oxidation, the biodiesel that will produce from these fats will need some sort of oxidative stabilizer to be used safely as a fuel. So, to summarize, the ideal substrate for biodiesel production would be low concentration of saturated fats, low concentration of polyunsaturated fats, and very high concentration of monounsaturated fats, which is basically what we are getting from the microbe. Low concentration of saturated fats, low concentration of PUFAs, and very high concentration of monounsaturated or oleic acid. And when I said in the introduction that this process is directly applicable to existing systems, this is what I meant. This composition is very similar to the soybean and the trunca oil, which is currently being used in the US and the European Union for biodiesel production. So we can directly substitute microbial fats by these fats. And now let's look at the conclusions and implications of our study. So what I just presented represents a very novel and extremely flexible technology which can take organic waste from diverse sources, from multiple sources, and channel them to just one endpoint biodiesel. And since we do not stress upon any agricultural commodities in this process, this biodiesel production technique is actually a step away from the whole food versus fuel debate. And this is highly sustainable. And we noticed that when we combine this process with in situ extraction, the extraction of the lipids inherently present in the waste, we can increase the final biodiesel yield by as much as 40% in the yield. And now finally, last thing, we can do all this using an algal biodiesel platform, but this is more lucrative since we can also provide sanitation using this process. Thank you.
Thank you, Shashwat. We have time for some questions. If you can come up to the microphone and state your name and ask the question.
Nicholas Ashmont, University of Alberta. Question is the competitiveness of your yeast, given that these waste streams come with a lot of other bacterial biomass. So either do you have to pre-treat to suppress the bacterial activity, competitors, particularly to produce some of these fatty acids and lipids, or is your organism very competitive in mixed culture?
Yeah, well, organism is competitive, yes, but the growth conditions for yeast and bacteria are very different. So this yeast grows at a pH of 5.5 and 6, and the influent stream contains very high volatile fatty acids, which is not ideal for bacterial growth. The yeast is highly competitive when compared to bacteria under those conditions, at a low pH and a high acid concentration.
I was thinking more of the acetic acid bacteria, those sorts of organisms that would thrive in those conditions.
Well, we haven't really looked at mixed culture of yeast so far, which is pending studies, and we'll work on that in the future.
Thank you.
I have a question. So in your batch or chemostat studies, under ideal conditions of CRE to N and so on and so forth, the metabolic modeling, how much of the carbon is converted to the lipids? And how much is converted to biomass and how much would stay?
Compared to that,
Best case scenario.
so I did not do this slide, it actually had this. This is from the chemostat, and we were actually able to get almost 30% intracellular lipid concentration and almost complete removal of VFA. And infant VFA was at 5,000 mg per kilo, 6,500 mg per liter of COD. And we were able to get almost complete removal of all the COD inside the body, inside the chemostat. So this is the maximum we achieved, almost 30%.
Did you try different HRTs on the chemostat, or it was you optimized
Not
between
yet,
it?
since I'll have to go back to see the effect of nitrogen concentration. Under the low growth conditions, the cell tends to produce more lipids, and therefore we were operating at almost 1/3 of the maximum cell growth.
Dan Noguera, University of Wisconsin. Is the lipid just stored in the membrane, or is it excreted, or in salmonella inclusions, or what?
Well, the membranes are phospholipids, so of course it's in the membranes, but also the intracellular lipid content increases. It's not excreted, it's not extracellular, it's either in the membranes or intracellular.
More questions? Okay, if not, right here.
Have you developed a method for extracting the lipid once you've produced it?
Yeah, so we were doing it at a lab scale, so we were using existing technologies, but at a commercial scale, soft cell extraction has been used to extract lipids from the biomass using chloroform.
Solid extraction.
Yes, solid extraction.
Okay, very well then. Thank you, Shashwat. Let's give it one more time. We have a couple of minutes. I'm going to